So, good morning, everyone. Today we will discuss about chromosomal theory of inheritance. So, I given a caption that so one can link the traits to the specific chromosomes. So, how one can say that the genes controlling particular traits are located on particular chromosome. So, this is what is a chromosomal theory of inheritance. So introduction. So after Mendel's work was rediscovered in 1900, so no possible relationship between genes and chromosome was established. So do you remember I was telling in the classroom that so even Mendel discovered or proposed two laws of inheritance. So he was not knowing about chromosomes, cells, and genes. So he simply called them as traits. So he was not knowing those traits or genes are located on chromosomes. So by 1910, many biologists suspected that genes were situated on chromosomes, but they did not had a proof. So in 1900, so many biologists they are thinking that genes could be located on chromosomes, but nobody had actual proof, a genetics proof to say that genes are located on chromosomes. But in 1910, an American biologist, Thomas and Morgan, published the results of an atypical male fruit fly that appeared in his laboratory. So this atypical male fruit fly had a white hide phenotype. This is unusual fly which he observed in his laboratory. So that is what we are going to discuss about that atypical fruit fly which had a white eye color. And so this Morgan was able to show that eye color in Drosophila melanogaster is physically located on X chromosome. So what he did was he showed that eye color in Drosophila melanogaster is having a gene which is located on a X chromosome. So by doing this experiment, so he linked that so eye color in Drosophila is located on X chromosomes. So after Th Morgan's experiment, so one of his students, Calvin B. Bridges, provided definite proof of chromosomal theory of inheritance or heredity. So we will discuss these two theories, means the experiment done by Th Morgan and the experiment done by his student Calvin B. Bridges. So for this, Th Morgan is the Th Morgan. Thomas and Morgan received a Nobel Prize in 1933 under physiology or medicine category. But unfortunately, he did not share his Nobel Prize with his student, Ph.D. student, C.B. Bridges, who gave actual theory of chromosomal theory of inheritance. In fact, he gave a full proof saying that genes are located on chromosomes. That is a sad part of this discovery. So next, why Drosophila is used as a model organism? So why not other organisms? So Drosophila is also known as syndrome of genetics. So this is the life cycle of Drosophila from adult to egg. And so there are four main reasons why a geneticist opt for this Drosophila melanogaster is it has short life cycle, means it has only 12 days from egg to adult, and ease of culture and maintenance. It is very easy to culture. If you just put a banana in a vial, and all these flies can be cultured and can be maintained very easily in a laboratory condition. And the next reason is that it has only 8 chromosomes. That means six autosomes, and one pair of allosomes. So that means three pairs, 6 chromosomes of autosomes, and XX in case of female, and XY in case of male are there. And the next one is the unique feature of this Drosophila is it has very small genome size in terms of bases. So if you take in terms of bases, it has very small genome. So the whole genome sequence of Drosophila melanogaster was deciphered in the year 2000. So the actual genome size of this Drosophila is 132 MB. That means 132 followed by 6 zeros, 132 million bases. If we count the base from first chromosome till this eighth chromosomes, it has 132 million bases. So it contains around 15,500 genes on these 4 chromosomes. That means if you compare the density of genes means the number of chromosomes in a given location. Sorry, number of genes in a given location is density of the genes. The density of the genes is more in human as compared to density of the genes in Drosophila melanogaster is more as compared to human genome. So why I am telling is that the human genome has 35,000 genes, but the actual size of the human genome is 3.2 GB. That means 3,200 MB. The genome size is more, the number of genes are less. So genes are sparsely dispersed. So in case of Drosophila, the genome size is less, but it has 15,000 genes. These 15,000 genes are densely located. So what Th Morgan did was actually so when they were doing this experiment simultaneously, like in Mendel, so they were trying to Mendel discovered traits, so very simple traits to be studied. So however, this Th Morgan and his students in Columbia University in United States of America, so they were trying to figure out or identify a unique trait in Drosophila melanogaster to study this genetics. So however, they could able to detect a fly which has white eye phenotype. So usually if you observe the eye color of this Drosophila melanogaster, it has red eye. But there was these people, Th Morgan and his student, they were hunting, searching for a trait which is very, very contrasting to study the genetics. So fortunately, they detected a mutant male fly that had white eyes. So white eyes, which is a recessive trait as compared to red eyes. So red eyes is the dominant trait. So you know that dominant trait is the one which surpasses the mutant or recessive allele in the F1. So what he did was, so these people like Th Morgan and students crossed red-eyed female. So red-eyed female means it is a wild type female, and crossed with an unusual fly they identified in the laboratory, that is white-eyed male. So this is a female which has homozygous wild type. That means it has a red eye phenotype. So I have represented as XX. So I told females carry XX, males carry XY. It is like human beings. So the gene responsible for eye color located on X chromosome is represented by plus symbol. So plus symbol means we always represent the wild type alleles as plus. And this homozygous wild type female was crossed to a mutant male, which is carrying white eye phenotype. So this was the fly they identified in their laboratory. So which has white eye phenotype. Look at the eyes; it is white. So this is the typical, atypical male fly they identified. So what they did is they picked up these females which had a red eyes, and they crossed to this atypical male which has white eyes. So these are the gametes, these are the eggs this female can produce. Eggs carrying X plus means allele for red eye phenotype and allele for red eye phenotype. So this carries only one kind of, produces one kind of eggs. So it doesn't mean that it doesn't have another 3 chromosomes. Those 3 chromosomes are autosomes. It carries 3 plus this X chromosomes. This X carries 3 autosomes, and along with autosomes it carries this X chromosomes. So similarly, this male, white male produces um, sperms which carries a mutant allele, that is recessive allele, which is responsible for white eye color, and the one more kind of egg which carries Y chromosomes, wherein which the genes controlling this white eye phenotype is not located. So with this assumption, so when this sperms fuse with this egg, it gives X plus and XW genotype. Similarly, this um, sperm fuses with this egg, it gives X plus and XW. And similarly, this sperm fuses with this egg, it carries X plus and Y chromosome. And similarly, this sperm fuses with this egg, it carries X plus Y. So just if you look at this plus W plus and Y and plus and Y, so these are the kinds of flies like phenotype we can expect. So what is the phenotype of these flies? So when you produce this homozygous female and a mutant male, so they got all the F1s, means all the progenies have a red eye phenotype. So this is the eye, red eye. So it has multiple facets or small eyes called like small eyes. So all of these small eyes are made into a compound eyes. This is the eye. So the germ structure of this eye. So that means when they cross this homozygous wild type female with unusual fly, white male fly, so all the progenies were red eye. So what you can see is, so that white eye phenotype is disappeared in this generation. So if you categorize these flies into male and female, so these top two flies are females and bottom two flies are males. So that means 50% of the generation, 50% of the progenies are male and another 50% of the progenies are females. But white eye color was disappeared. As I already told, the white eyes are recessive to red eyes. So they were surprised. So the parent had white eye character, but the progenies did not had white eye characters. So what they did was, this is one more thing. Just remember, I wanted to explain the crisscross inheritance. So this is the daughter. This female is a daughter to these parents. So this daughter inherit X chromosomes from their mother. This red X chromosome is from their mother, and green X chromosome, which is carrying white eye allele, is from a father. So why it is this daughter is why it is red color? Because the wild type allele is received from their mother. So similarly, so all these sons, like these males, will become sons to these two parents. So all these sons will inherit X chromosomes. That's just track of this color, red color X and red color X. This red color X chromosome was inherited from the female. That means a mother. Whereas you know that all the sons will receive the Y chromosomes from their father. And why it is red? Why the sons are red? Because they received the allele from the mother. So mother carried a wild type allele, which is responsible for red eye phenotypes. So because of this, like in almost all the progenies, the X chromosome carrying wild type allele, that is plus, contributed to all the progenies. Because of this reason, all the progenies were red, and and this character was disappeared. But just observe this first two, females, which had green X and green X, which are carrying the recessive allele for white eye mutant. So this was surpassed, this was masked by this dominant allele. So they were shocked to see that all the progenies were red. But however, what they did was, so they took this heterozygous female X plus and XW. This is an heterozygote, and X plus Y as a male. So they took these females and they took these males and they intercrossed this. The next experiment Th. Morgan did was he crossed heterozygous individuals. So this is the female, means a daughter, which is carrying X plus and XW, and this is the son, which is carrying X plus and Y. So this is just like a brother and a sister mating. So this is just a brother and sister mating. These kind of matings are possible only in animals and only in animals and these insects, except human beings. In human beings, we cannot do this kind of experiments because of ethical reasons. Whereas this Th. Morgan pick up these females, daughters, which are heterozygous, and sons are the male flies, and they intercross. So when they intercross, so they got four different kinds of a genotype. That observe plus plus, plus W, plus Y, and WY. So four different kinds of genotype. So what was their phenotype? So what was the external appearance of these flies? Means, so out of this four different kinds of genotypes, so these progenies had red eyes again. So these progenies had red eyes. So and this progeny had white eyes. That means they recovered this white eye color phenotype in the F2 generation. So so if you categorize these progenies, so it doesn't mean that it produces only four progenies. It will produce these female lies thousands of eggs when they hatch out. So if you categorize, so there could be like 250 this kind, 250 this kind, and 250 like this, and 250 like this. So there will be only four kinds of genotypes and two kinds of phenotype. Two kinds of phenotype means red eye flies and the white eye flies. So four kinds of genotypes means it is plus plus, plus W, plus Y, and WY. So please understand this concept. Okay. So if you categorize based on the phenotypic observation, means the eye color, the 75%, see 75% of the flies are red, and only 25%, which I have highlighted in this circle, like dotted circle. So only 25% of the flies were white. So again, if you made a one more categorization or classification based on their sex of the flies, that means 50%, the top two kinds of flies, the female and female, means 50% of the flies are red. That means to say all the females will be red. Whereas if you take a male sex, the 25% or 50% of the males are red, and another 50 is a female. So if you take the entire thing, so 50 female red, 25 male red, and 25 male white. So what they did was, so based on this, they concluded that, the gene responsible for eye color in Drosophila is located on X chromosome. So yeah, the X chromosomes. But they wanted a validation, like for confirmation they wanted. So what he did, Morgan carried out an additional experiment to confirm this hypothesis, saying that the eye color gene is located on X chromosome. So what he did was, he took two approaches. One is he crossed heterozygous females and hemizygous mutant fly. So this is heterozygous plus and W. So it is a red-eyed female he took, and he crossed to a hemizygous mutant male. Hemizygous means which carries only one allele for the eye color. So that's why it is called as hemizygous. That means including human beings or the animals or the anything which carries XY chromosomes, we are having hemizygous situation. That means we carry only one allele for the particular triad, whereas females carries two alleles for the particular triad. So these are the hemizygous males. So it was hemizygous for white eye, hemizygous for eye color. So when they cross red-eyed female with white-eyed male, so they got these kind of progenies, four different kinds of progenies. So red-eyed female, white-eyed female, red-eyed male, and white-eyed male. And so that means if you categorize these progenies based on their sex, so 50% of the sex had white eyes and another 50 had red eyes. So one more approach or one more experiment, what he did was, he crossed a homozygous mutant female with hemizygous wild type male. So this is a homozygous. This female is white. So whereas in this experiment he took red-eyed female. Whereas in this experiment he took white-eyed female. So white-eyed female was crossed to a wild type male. Wild type male means a male which carries a red-eyed phenotype. So when he crossed, he got a two different kinds of flies. One is red-eyed female, another one is white-eyed male. So two different kinds of F1s. So what he did was, he intercrossed this. So again, these are like brothers and sisters. They intermated these two progenies, and she got a red-eyed female, white-eyed female, red-eyed male, and white-eyed male. So the end results of this and this experiment is same. So based on all these experiments and validation, so Th. Morgan concluded that eye color gene is located on X chromosome. So this is very, very important a topic or the theory proposed by Th. Morgan to say that to simply say that genes are located on chromosomes. So next, what happens is, I told it was like full proof concept or the validation was given by student uh, CB Bridge. So what they did was, when they did the thousands of matings, they did thousands of matings to predict that the white-eyed flies in D.H. Morgan's experiment, the rare occasion, they discovered exceptional white-eyed females. See, the first Th. Morgan's experiment was based on a white-eyed male. But when they were doing this white-eyed, the previous experiment on white-eyed male, they exceptionally discovered unusual, like again atypical white-eyed females. So previous experiment, white-eyed males, but one more experiment, when they were doing experiment on these white-eyed males, they discovered an exceptional white-eyed females. So this observation was made by none other than his wife, Lillian von Morgan. So Th. Morgan's wife's name is Lillian von Morgan. So she recognized these unusual flies which had a white eye females. So according to her, these exceptional females might have an unusual chromosome composition, that is XXY. So indeed, these females had two X chromosomes as well as one Y chromosomes. So why this carrying three sets, two X chromosome and one Y chromosome, is because of the abnormality during the gamete formation. So according to her, she says that these XXY females are because of high frequency of non-disjunction of these chromosomes. So what is non-disjunction? So we'll discuss in the next slide. Is the failure of chromatids to separate during the second meiotic division. So this is the experiment, second theory proposed by CB Bridge, Calvin Blackman Bridges. So his theory's name is non-disjunction as proof of chromosome theory of heredity. So from 1913 to 16, CB Bridge performed his experiments to say that genes are located on chromosome. So in fact, whatever the non-disjunction theory proposed was his doctoral thesis, a research problem. So he did his PhD under the guidance of Th. Morgan in Columbia University, that is in New York City in United States of America. So in this experiment, what he did was, so he studied on Drosophila, which is a common fruit fly. So he showed that a process of a non-disjunction. So non-disjunction is described by Bridges causes the sperm and egg cell to contain abnormal amounts of chromosomes. So normal chromosomes was supposed to carry 3 autosomes, 1 sex chromosomes, means 4 chromosomes. So, but this, because of non-disjunction phenomena, so these eggs and sperms carry abnormal quantity or amount of X chromosomes. So what is non-disjunction? So non-disjunction is the failure of pair of chromosomes. So failure of pair of chromosomes to separate during So this is what he took white-eyed female, CB Bridge took white-eyed female and red-eyed male, and again I have taken XX, which female flies carry XX chromosomes, and male carries XY chromosomes. So this is the normal eggs produced by females. So normal eggs means it carries only one X chromosome along with 3 autosomes. And these are the sperms which are produced by this kind of males, which carries sperm carries X chromosome and sperm carries Y chromosome. So in the normal process, when this egg fuses with this sperm, so it gives XX flies which are females. Okay, these are having red-eyed phenotype. When these sperm fuse with this kind of eggs, it can XY. So because it is XY, it is a male, but it carries a W allele, that is a recessive allele, which codes for white-eyed male. So a CV Bridges experiment concentrated on non-disjunctional theory. So what it does was, this female will produces the eggs. So, okay, it produces the eggs which are having both the X chromosomes. See, this chromosome was supposed to go to this sex, but it will not go because of failure of separation. So because of these both the chromosomes will go to one egg. So these eggs will fuse with this kind of sperm. So it gives triple X, XXY, a female. And this sperm with this nulloy X, but nulloy X means it doesn't have a sex chromosome, but it carries autosomes. So it gives only a X0 flies. And this sperm fused with this egg gives XXY, and this egg fused with this egg gives a Y chromosome. So usually this Y0 flies will die. I will tell you the reason. And also this XX, triple X, meta female, these are known as meta female. These are also dies. The only thing is exceptional red-eyed males which have X0 and exceptional white-eyed female which are having double X, XXY, exceptional females. So these are the flies which were recognized by Th. Morgan's 5. So for this, CB Bridges is giving an experimental proof. So based on this experiment, they observed that exceptional white-eyed females were XXY, and the exceptional red-eyed males were X0. So these are exceptional red-eyed males were like X0. So since X0 flies were male, he concluded that in Drosophila, Y chromosome has nothing to do with determination of sexual phenotype. You see, the X0, so it doesn't carry one more X chromosome, but these flies will survive. But these flies look like these flies are male. See, Y chromosome is not there. Still, their sex is Y. So according to this, so he says that Y chromosome is nothing to do with a sexual phenotype or determination of sex in Drosophila. And but in X0, here the X0 males were sterile. So these X0 males were sterile. So he realized that chromosomes must be important. That means X chromosome must be very important for male sexual function. So at least one X chromosome is needed for viability. So this one, so Y0 flies will die. Okay, but these flies will die because it doesn't have X chromosome. Because of this, he says that minimum one X chromosome is required for viability of the flies. So in addition to proposing the non-disjunction theory by C.B. Bridges, so he gives all these concepts. What is the importance of this X chromosome? What is the importance of Y chromosomes? Why these triple X females will die, and all those things. So according to, based on this, he proposes one more theory called genic balance theory of sex determination in Drosophila, but I am not going to deal in this course because so we have a separate chapter in next course, that is in genetics. So according, just in brief, what is genic balance theory of sex determination in Drosophila is, it is the ratio of X chromosomes to number of autosomes. So how many X chromosomes divided by how many autosomes. So that gives the ratio. So based on this ratio, the sex of the individuals are classified. So if the ratio of X chromosome to autosomes is 1, it is female, and 0.5 is male. These are normal. If it is 1 to 0.5, if the ratio is 1 to 0.5, it is intersex. If it is more than 1, meta female, and less than 0.5, it is meta female. And these meta male are sterile and weak. So I'm not going to discuss in detail about all those things. So in addition to proposing the non-disjunction theory of chromosomal inheritance, he gives all these concepts. C.B. Bridges gives all these concepts. So further evidence that genes are located on chromosomes, like whatever Th. Morgan did, C.B. Bridges gave a full proof concept to say that genes are located on chromosomes. And he concluded that sex is determined in Drosophila by the X chromosome and not the Y chromosome. This is very, very important point. So sex chromosome in Drosophila is determined by X chromosome. I'm stressing X chromosome only, not the Y chromosome. And the third concept he gave is he found that the presence of Y chromosome did not determine sex. Rather, number of X chromosomes did. Means the mere presence of Y chromosomes will not determine the sex of the Drosophila. Instead, the number of X chromosomes will decide the sex of the Drosophila. And one more concept he gave was, so minimum one X chromosome is very, very essential for the viability of the fly is concerned. So minimum one X chromosome is enough or essential for the survivability of the flies. So even flies carry more number of Y chromosomes, but no X chromosomes, or no Y chromosomes, all the flies will die. So this is what is about non-disjunction theory of chromosomal inheritance proposed by C.B. Bridges. These two theories, like T.H. Morgan's experiment on white-eyed male and C.B. Bridges' experiment on white-eyed female, like exceptional females, gave a foolproof concept to say that genes are located on chromosome. So I have two more concepts. These are very, very simple, like chromosomal basis of Mendel's principal segregation, to says that the chromosomes are located on genes are located on chromosomes. I will share you the PPT. So it is very simple because I have only 3 minutes to close this chapter. This is very simple. You go through this. If you have any problem, you can message me, or whenever we meet, we can discuss. So just I have taken monohybrid experiment to say that alleles segregate. And one more thing is chromosomal basis of Mendel's principle of independent assortment. That is second law of Mendel. By taking second law of Mendel, we can say that so genes are located on chromosomes. How the dihybrid ratios or dihybrid crosses can be taken to explain the genes are located on chromosomes. These are the gametes. So you know that it produces, if you take the F1, it produces 4 different kinds of gametes. So this is all about chromosomal theory of inheritance. The crux of this experiment, or crux of this topic, is to say that just genes are located on a chromosome. So that is what T.H. Morgan and C.B. Bridges, his Ph.D. thesis, gave. So thank you so much.